Hello everyone, welcome back to the Science Shack for another video in the photosynthesis series. Apologies if you can hear any screaming and yelling outside the back of the Science Shack. That seems to be a gaggle of science children screaming around the science bunker, so I can only apologise for them. Right, so this is the third lesson in this series, which is all about the light-independent reactions. You should already have watched the ones on chlorophyll, structure and function, and the one on the light-dependent reaction, so the bits that involve light, splitting water, and you should know what is produced. So, to crack straight on, same thing applies as last time, the three levels. We're going to present what I refer to as the TOMI Early Learning Centre, my first light-independent reaction level. Then one I think makes a little bit more sense because it's the numbers in this lesson that are problematic, and then maybe a little peek over the wall, a little peek at a bit more detail in the full sort of more detailed version. However, that is entirely unnecessary, so I may omit that by the time we get to the end. Okay? So in terms of context, it's always important to think about where we are inside the cell. Fundamentally with molecular biology and biochemistry, I think people only get lost because they forget the big picture of what it is that we're talking about. In this case, what it is that we're talking about is a leaf with a leaf cell, with a chloroplast, and this time we're based in the gooey bit outside the thylakoid called the stroma. Right, we're based in the stroma. Okay, so last time we were here in our thylakoid membranes, on our thylakoid membranes, building proton gradients and chemiosmosis. Now this time, all this chemistry is taking place here in the stroma. So there is an absolutely crucial enzyme in this, which is called Rubisco. Okay, and this is as good a place to start as any, because as you will see, this is a cycle. Okay, now Rubisco stands for ribulose bisphosphate carboxylase oxidase, which is a fantastic thing to scream at people as you jump out of an airing cupboard at them on Halloween, it being that time of year. Take it from me, it'll get the job done. So, ribulose bisphosphate carboxylase oxidase is a crucial enzyme, and it is one of the most important proteins on the planet. I know teachers are prone to hyperbole, but without this, everything else dies. Okay, so, however, it's rubbish. Okay, its job, its crucial job, is to fix carbon dioxide into some kind of organic form. Okay, so we dealt with the water and the oxygen in your GCSE understanding of photosynthesis last time. This time, it's the carbon dioxide and the sugar we have to deal with. So at some point, that inorganic carbon dioxide has to be incorporated into organic molecules and become part of glucose. And the first step that makes that bridge from the inorganic to the organic is mediated by this enzyme, Rubisco. Okay, and it is a terrible enzyme. So it's a really good opportunity for examiners to ask you questions on enzyme action, linked to photosynthesis, linked to evolution, linked to inhibition, and all that kind of stuff. So all these kind of areas of the A-level syllabus linked together around Rubisco. So it's worth knowing a little something about. To give you an idea of how rubbish it is, typically enzymes can work at thousands of reactions a second in very, very low concentrations. They can work in isolation. They don't need any help. It is all grand. Okay. However, this one, by comparison, 3 reactions a second. Typical enzymes we can give out to students in a classroom. You've probably done this. They can do experiments with them, drop them in dropping tiles and make starch get digested into sugar and do tests on them within a few minutes in the middle of an hour-long lesson. Rubisco is thousands of times slower than that. It is a hopeless enzyme. It needs hundreds of helper proteins that are more modern than Rubisco, that have evolved after it, to funnel it towards its right substrate, okay, to help it find its right substrate. It's, as a result, right, and this is one of the conditions you should know about enzymes, the cells, or the chloroplast, has got millions and millions and millions of copies of this enzyme, because by increasing the enzyme concentration, you increase the rate of reaction. Okay, fascinating figure, right, that I heard, is that if you were to dry out a chloroplast and measure all of the components by mass, over half the mass of a chloroplast, or half the dry mass of a chloroplast, is just this enzyme. Think about that. That means there is more of this enzyme by mass in every chloroplast than there is chlorophyll, than there are membranes, than there is ATP synthase, all because it is so hopeless at its job, okay? It occasionally as well can't even do what it's supposed to do correctly. What it's supposed to do is bind a chemical called RUBP, or ribulose bisphosphate, to carbon dioxide, okay? And that's the fixing step. I'll get to in a moment. Sometimes, however, it binds oxygen on there instead, right? And that is not great. It takes a lot of ATP to recover that. So on a lovely sunny summer's day, when there's plenty of water, there's plenty of carbon dioxide around, it's plenty warm, you know, all that kind of stuff. There's plenty of light for a plant. The limiting factor to photosynthesis is the efficacy of this enzyme, which is absolutely staggering, my friend. Absolutely staggering. So why? Why is it so rubbish? Well, it's because, in a way, it's so important, okay? It's derived from really ancient chloroplast DNA, okay? It evolved billions of years ago to do this job, and there's been no selection pressure to do any better. Bear in mind, if there was somebody designing this, you'd never design it, or you'd go back to the drawing board and you'd redesign a new standard enzyme to do this. And this is something that genetic engineers are trying to do, is improve Rubisco. So there's another syllabus link between this and the genetic technology, biotechnology part of the course that you haven't studied yet. There is a series on that, on mystical science stuff, so go and have a look at that if you're interested in biotech stuff. Okay, so plant cells, as a result, evolution only ever moves forward, have never developed or selected for a better mutation, because basically they get by. The plant compensates by making more copies, it survives, it passes on its genes, and that's fine. So basically the lesson evolutionarily here is that life ain't perfect, but just enough is good enough. Okay? So let's see what that Rubisco does. Now this is, I say, Tomi's Early Learning Centre, my first Calvin cycle, as it's called. The light independent reaction is a cycle. It is called the Calvin cycle. And basically, it is the way, in a cyclical fashion, that carbon dioxide up here is incorporated into complex molecules that are spat out down here. Okay? Now, those of you who are paying proper attention will be confused in a minute. If you are confused as to the numbers not adding up or it not seeming to make sense, good. That means you are paying attention. It doesn't mean you're not understanding, it means you are understanding. This is a simplification, and I will take the level up a little bit in a while. Okay? So, first thing that happens is we take a 5-carbon molecule called ribulose bisphosphate, and this enzyme, Rubisco here we've just talked about, combines that with 1 carbon dioxide. Okay? That makes a 6-carbon compound. Obviously, 1 carbon, 5 carbons, 6-carbon compound. Okay? That 6-carbon compound isn't even mentioned here because it is so unstable that it immediately breaks down into 2 molecules of glycerate phosphate, or glycerate 3-phosphate. Okay? So that's 6 carbons in total. Okay? Now here is where we need these things, which you should recognize from the last video. Okay? This reaction to the next stage is endothermic. It requires an input of energy and it requires an input of a proton to do so. Okay? So the ATP that we made in the light dependent reaction is used here along with the reduced NADP. Okay, both its energy and its proton, electron, are used to turn this 2 molecules of glycerate phosphate into 2 molecules of triose phosphate. Okay, now triose phosphate, as we'll learn, is a hugely important molecule in lots and lots of metabolism. It's a massively important intermediary in all sorts of different processes. Okay, so this is endothermic. The ATP and the ATP becomes ADP. The reduced NADP, sorry for the science children, and the becomes NADP. And these, the ADP, the PI, and the NADP, are recycled back into the light-dependent reaction. Now, an examiner's favourite trick here is to go, okay, what happens, or words to the effect of, what happens if you turn the lights off? Okay? To look at some exam questions later where we tweak different things like the CO2 and the light. And the answer to that question is, well, this in and of itself wouldn't be affected by the light being turned off, but you would not have any ATP or reduced NADP. So you would build up glycerate phosphate. Okay? That's what would happen. Now, triose phosphate, and this is the bit where we all have to kind of just go, RUBP. Right? That's basically what we're talking about. Okay, so 2 triose phosphates here. There's a series of lots and lots and lots of complicated chemical reactions that you do not have to know about for A-level purposes, right, require a molecule of ATP over here to turn the triose phosphates back into RUBP. Okay, and obviously what's left over out of this is this extra one carbon. Now you might be looking at that and going, well, how can one carbon make complex molecules? That's basically, we've got one carbon over here, so what's this? And if you're thinking that, good, well done, you have understood. This is a simplification. Okay, so to go through one more time, we take our 5-carbon ribulose bisphosphate, we combine it with our 1-carbon carbon dioxide. The enzyme that does that is ribulose bisphosphate carboxylase oxidase. That creates an unstable 6-carbon compound, which breaks down into 2 glycerate phosphates almost immediately. That then requires an input of energy from the light-dependent reaction in the form of ATP and reduced NADP. Okay, round we go. That creates triose phosphate. Triose phosphate is crucially important in manufacturing complex molecules like glucose and ribose, glycerol, and all these other wonderful things. Then via a series of things, RUBP comes back, which is 5 carbons, and that requires an ATP as well. Okay, so it shouldn't take a genius to answer the question, how many times round this cycle does this cycle have to turn to get one whole glucose? You should know glucose is a hexose sugar, meaning it requires 6 carbons, okay, and those 6 carbons come from 6 turns of this cycle. Now the bit that obviously, if you're looking at that thinking the numbers don't work, the bit that we've missed out is this doesn't happen in isolation, right? One carbon dioxide is not going in on its own. We can't think of this in terms of one carbon dioxide. So let's go up from the early learning centre, Tomi, my first Calvin cycle, up to the Meccano lesson. I think that's probably where we will get to. And I can give you some links in the description if you want to go any further than this. Okay, so this link is much better. Okay, and I'll show you what the numbers work out, because at least this diagram acknowledges that there's another step there, our 6 carbon. Okay, and there's also a whole bunch of steps here. It's not actually just complex molecules that get taken off. Every 3rd cycle, we've effectively generated another triose phosphate. Now that triose phosphate can be made into all sorts of things. Primarily for the understanding of your GCSE, to make your GCSE understanding of photosynthesis make sense, the thing we're making is glucose. And then glucose can be turned into sucrose, starch, cellulose, amino acids, lipids. Obviously for amino acids we have to add a source of nitrogen, nitrates. You can look back at the other cycles chapters to get an idea of that. Okay, so we need to think about this whole cycle, okay, in terms of multiples. Right, the minimum we can think of in terms of is 3s. Okay, so if we didn't have 3 ribulose bisphosphates here, okay, then what we would have is if we had 3 here. Apologies, just my colleague joining me there for a moment. So I'll start that bit again. If we had 3 5-carbon molecules, okay, then what we've got is 15 carbons here. Okay, so we've got 3 ribulose bisphosphates, and if we've got 3 carbon dioxides, okay, then we can add those together and get 18 carbons in total. Okay, so that's 3 RUBPs and 3 carbon dioxides added together, right, to make 3 6-carbon molecules. As we said, those 6 carbon molecules are unstable, so they will break down and make 6 3-carbon molecules, 18 carbons in total. Okay, so that's this point here. We've got 18 carbons in total, right? So then we've got 2 ATP, 2 ADP and PI, right? That will obviously need to sort of triple that up as well. So we need more ATP, more NADPH, and that makes us our 6 triose phosphates. Instead of having 1 and 2, we've got 3 and 6. Okay, so 6 triose phosphates. Now that being 18 carbons, we can then take 1 triose phosphate away to leave a pool of 5 triose phosphates, and those 5 times 3 is 15. Okay, then we've got the right number of carbons to put a little bit of energy in, right, and regenerate 3 5-carbon RUBPs. Okay, so that's how the numbers make sense. You can't think of it in terms of single carbons, single moles of carbons. You've got to think of it in terms of multiples. So for every 3 turns you get a spare trilose phosphate, for every 6 turns you get a glucose. Okay, so that's quite important, and I think this diagram even explains that further. So we've got our 3 5-carbon molecules here, 3 single carbons, our temporary 3 6-carbon molecules, which break down into 6 lots of glycerate 3-phosphate, which are 3 carbons. Then we put our energy in, our ATP and our NADP, H, that goes in there. We've got our triose phosphate, 6 lots of those, okay, and one of those can be taken off. Okay, one of those can be taken off, leaving us with 5 3-carbon compounds, which can be regenerated into 3 5-carbon compounds. Now there are a lot of steps in between here and here, okay, but that would be the full detail, the full actually what happens. I don't think for the scope of this video we want to go